We will be together in the next about 2 weeks, and you will not see every day me, but you will meet many people from our team, the brain simulation section at the Charité University Hospital in Berlin, Germany, and the Berlin Institute of Health. And specifically, you will meet these people, so quite many from our team, who will give you insights in the work and experience we have in the field of brain digital twins. I'm sure you have seen the agenda. I just want at the beginning to explain it to you once more. So we have 2 weeks together from today until Tuesday, June 30th. And all the sessions will start at 4:30 Central European Summertime and will go for 2 hours, except for Friday. There we will start a little bit later because one of our lecturer is actually a medical doctor, also active on the ward with patients, and therefore we have to move a little bit the lecture to later so that he can first finish with the patients and then he will be available for you. So let's have a look at the agenda. You find it on our website. I will share the link once more. Okay, and again, so we record the lecture, but we prefer not to take automated notes in the cloud because the notes would also contain all the interactions of the attending people, and this is not desired for data privacy reasons. So please do not start your automated note takers. The link that I just provided brings you to our website and to the entire program. You will see there the topics and the names of the lecturers. So today I will give an overview of the virtual brain. The virtual brain is a simulation framework, and a lot of scientific work has been done internationally with this whole brain simulation framework. I will give you a couple of examples today and hopefully inspire you so that you are getting curious and want to use it yourself. And if we screen a little bit through the program, you will see that it's really very rich. So the more technical introduction and the topic of brain network simulation and how this can be done in a reproducible and also automatable way will be provided by Leon Martin tomorrow already. Then we will also have on this Wednesday a very important topic provided by Michael Schörner and Mark Sachs, namely GDPR compliance. Yeah? Not only in Europe, also in other regions of the world. Data privacy is important, and there are different legislation. In Europe, this is the General Data Protection Regulation. If we deal with digital brain twins, then we have to also protect the privacy of the individuals whose brains we are simulating, and how this can be done will be explained on Wednesday. And Michael Schörner and Luca, who I have also seen online today, will give a lecture on digital twins and intelligence and plasticity. They will show the work that is done in the brain simulation section on these topics. Then we have on June 5th, on June 19th, I mixed the dates, so it's June 19th, of course, June 5th is in the past. So session 5, it is, we have the already mentioned presentation by Leon Stefanowski. He introduces a tool that he has developed together with other colleagues, where we, in a more automated way, extract knowledge from the available literature and include this automatically extracted knowledge in our brain network models to make the models more biologically realistic. Then we have the weekend and continue on Monday, June 22nd. There we have the topic generating virtual brains using tailored brain image processing workflows. And we will dedicate some focus to quality assurance. If we generate virtual brains, individualized virtual brains, then we have to first acquire data from the patients, from the subjects, and we have to process this data. And it is highly important to make sure that the data quality is sufficient to also ensure reliable and robust results. Then on June 23rd, we have the topic brain network model optimization and multi-scale co-simulation, where Marius Pille and Dionysios Pedikis provide insights in the latest developments in terms of parameter estimation, parameter search. So how do you infer a model, and how can this be automated and made most efficient? Then on June 24th, we have a session provided by Simon Lagunas and Jihai An on understanding principles of brain function in the mouse model. So this will be a session that is not dealing with human brain models, but with mouse brain models. Then on June 25th, Jill Meyer and Tiago Falicetti will share their experience with simulating brain stimulation, be it deep brain stimulation or transcranial brain stimulation. Then on June 26th, Chin Liu, Leon Deger, and Valerie Bragin will provide a lecture on modeling mechanisms of consciousness. On June 29th, okay, I see there is a note taker request, which I declined. On June 29th, Konstantin Bühlau and Christoph Hüttel will talk about modeling mechanisms of psychosis. And then in our last session on June 30, we will, or Halgor Taher, I should say, Jennifer Thiem and Rico Schmidt will dedicate the 2 hours to TVB, which stands for the virtual brain positron emission tomography, and in silico drug discovery and testing. So it's really a wide range of topics, but all, of course, linked to whole brain network modeling. And we hope to inspire you and to bring your interest in the topic to the awakening. In the last session, we also will provide more information about the final test. There will be a final test, of course, only for those who are interested to take it. And this will be circulated. There will be questions that we ask you then to answer and to send us back the answers. And depending on the correctness of the answers, you will pass or not pass. And then if you are not passing, you will get a second opportunity to pass the test. And if you have passed the test, then you will get the credits and the certificate for the course. So this is the plan. If you have not already downloaded the PDF with all the detailed information, then you can do this. You will also find it on the same website that I have shared before. So at the bottom, you will find the PDF, and there all the information is available. And feel free to also share the course with your friends and colleagues. So we are open, and as you know, we are also free, and we are very happy actually to reach our colleagues, other scientists and students all over the world, and to give the opportunity to take part in our expertise that we are very happy to share. As also mentioned already, the lectures will be available. In most cases, there can be one or the other exceptions, but in most cases, we will make the lectures available. We record them as we do right now. And we have a YouTube channel. There you will also find previous presentations from previous courses. where then for this course will be a new playlist, and there you will find all the recordings. And just put the YouTube link also in the chat. So I start with my own virtual brain twin. So you can see that it's really personal. This is all about personalized subject specific brain network, whole brain modeling. Here you see a simulation of my electroencephalogram. Then here my resting state, brain activity on the cortical surface. And these are really my deep brain structures, deep nucleus, cerebellum, that have been reconstructed from my own data. And this model that you just have seen has been constructed many years ago. We have by now many different kinds of resolution, depends always on the question that we want to address with our digital brain twins, but we can build now multi-scale models where parts of the brain really have a lot of more detail than what you have seen right now. I will show you a couple of examples for this also later on, to be able to capture what is actually of interest for scientific or also clinical question. So today I give you an overview, and then during the course of the summer school, you will go in depth with all the different scientists who have already worked on the topics. I will today show you some recent studies, some in more depth, some in less depth. So these are the 4 selected publications that I want to touch upon. First one shows you how static brain models can be already utilized for clinical decision making, but then quickly I move forwards to dynamic brain network models, as you can see here, and will demonstrate you how they can be useful in learning how the brain works, and also in using them for clinical decision making. So let's start with the first study. So funnily, all 4 have been published in the same journal in Nature Communications, but obviously by different authors and co-authors. So the first one has been created under the lead of PhD student Nandita Rayamani and Andy Horn. He's a senior author when we did this work. So he's actually a former colleague at the Charité. He then moved to Harvard, and now he is leading an institute in Cologne. And he has done a lot of work in the field of deep brain stimulation for different brain diseases. And this recent study that has been done as part of the PhD project of Nandita Rayamani has reconstructed the fibers in the brain based on diffusion tensor imaging, and developed a predictive model using these fiber reconstructions to predict for an individual patient which of these fibers need to be stimulated in order to best treat their symptoms in the case of motor disease. And this is, for example, Parkinson's disease, as you can see here in the title. So what you see here is a subthalamic nucleus and the globus pallidus internus and globus pallidus externus, the colored structures. And here you see also different colors. The fibers are colored according to their importance for treating different symptoms in Parkinson's disease. And these symptoms are tremor, bradykinesia, rigidity, and axial symptoms. Yeah, and depending on the dominance of one or several of these symptoms, different fibers and locations need to be stimulated via deep brain stimulation in the patient. And this has been retrospectively validated, and there's also already included in this publication a small prospective pilot trial. Yes, this was a study from 2024, and more work has been done since then. But as you probably will see, of course, this is a static model that does not take into account the dynamics, which are important, of course, for cognition and for brain function. And the virtual brain, the brain network modeling framework, can actually take into account how the different regions in the brain that are connected via these fiber tracts are interacting and influencing each other and generate dynamics. And this is the framework that we are using in the brain simulation section, brain network modeling, whole brain network modeling. We have later extended it to multi-scale whole brain network modeling, which enables us to understand the dynamics that are being generated in the brain and constrained by the structural connectome. So this is a study where I want to go a little bit more in depth that has been led by, at that time, PhD student Dominic Koller. He now already some time ago received his PhD, and he is just now preparing for, at the end of this month, moving back to Austria, where he will do another career path. But he had joined our lab for some time and did this fantastic work on traveling waves, where he investigated 776 virtual brains. The data have been obtained from the Human Connectome Project in the US, and was able to reveal how the structural static connectome gives rise to these waves that you can see here. Yeah, you see here these wave fronts that travel across different brain regions, and at a wave front, yeah, this can be lines, this can be also a tilted line or a circle. We have synchronization. Yeah, the state of the neurons is synchronized at the wavefronts, and therefore just so interesting for us to understand how these wavefronts are being generated in the brain, because synchronization of sometimes distant brain regions is the basis for learning, memory formation, and plasticity. This is just another example of a traveling wave, local field potentials, and the marmoset monkeys. This is area MT. This is an enlarged view of this area MT. And here you see also such a traveling wave that is traveling from the left upper corner to the right lower corner. And the edge length, as you can see here, is only 2 millimeters. So this is really a micro array recording, a very small area in the brain. Yes, this is 2 millimeters. And the waves that we can see here are relatively slow, between 0.1 to 0.6 meters per second, which also indicates, yeah, as also the area of distribution, that they are mediated by non-myelinated inter-areal connections within this area. But then we have also other traveling waves that can be much larger. This is a human brain, and these are also empirical recordings, electrocorticogram in patients with epilepsy. And this is actually done during sleep spindle activities. Sleep spindles are very important oscillations in the brain that occur during the sleep that are important for memory formation. And here we see that there's actually a counterclockwise moving traveling wave. Yeah? Sometimes it's a little bit difficult to spot, but probably you will see it. And this traveling wave moves a little bit faster with a speed of 2 to 5 meters per second. And the spatial extent and also this traveling speed indicates that the underlying neurons are myelinated neurons that can pass on information faster. Now what determines the direction of traveling waves, some work already quite some time ago has been done and investigated this. These are the early studies that have shown that isolated frequencies, so here you see the frequencies with different intrinsic frequency ranging here between 1 and 0.6 hertz, actually generate a phase shift. Here you see the phase shift. We see that from fast to slower frequencies, there's a phase shift, a phase progression. Yeah, here's a phase. And this leads to a traveling wave. So if you have a network with a frequency gradient, as here constructed, in this model that has been built by Ermentrout in 1998, and you implement a frequency gradient, then you will generate a phase shift, and with this a traveling wave that travels over the connected network nodes. This was one observation already quite some time ago. There was another parallel observation, completely unrelated, by another team, and they showed that these frequency gradients that were here artificially introduced in this model network can be also generated by changing the node degree. So if you have a network of coupled nodes or oscillators and you modify the in strength or the number of incoming connections, then this differential number of incoming connection modifies also the intrinsic frequency. So as you can see here, the higher the node degree, the higher the number of incoming connections or connection strengths of this node, the lower is the frequency. So by changing the node degree, it is possible to systematically modify the intrinsic frequency in a network of coupled oscillators. So taking this together, there's a mechanism, namely the node degree in a network, that systematically can create a gradient of frequencies, and a gradient of frequencies can lead to phase shifts, and with this to traveling waves. So now, being aware of this, Dominic Koller generated first a relatively simple network, a grid network of coupled oscillators. And he, as you can see here, so these are the oscillators, and they are coupled, and he implemented an in-strength gradient. As you can see here on the right side, the in-strength is higher than on the left side, as indicated by this color code. And the question was now, if we produce such an artificial in-strength gradient in a network of coupled oscillators, would this produce a traveling wave along the gradient of in-strength? So this has been tested. And indeed, in this simple simulation, very clearly visible, these traveling waves occur. And if we now change the in-strength gradient and distribute it randomly in the network, then we can also see there are traveling waves, but they are not directed as here. They are not going from left to right, but they are more heterogeneously distributed, going in many different directions. So this was actually confirming the hypothesis that the in-strengths or node strengths in the connectome in the brain could direct the traveling waves that we can observe in brains. So now the question is, do we have such in-strength gradients at all in our structural connectome? And this was subject to the next analysis by Dominic and the team. And also here, we used the data from the Human Connectome project that we obtained from the US. And we systematically partition the connectome with different parcellations. Here you see the Schäfer parcellation, a relatively fine parcellation of the brain. And independent of which parcellation has been chosen, the outcome was always the same, namely, indeed, there are in-strength gradients in the brain. Namely, in the orbitotemporal region, we have low in strength, and in the frontal and occipital regions, we have higher in strength. So there are gradients from temporal to frontal and to occipital. So now the question was, if we now use such a structural connectome that has these in-strength gradients, would such a structural connectome produce traveling waves? And now we use these connectomes and hypothesize that once we construct a brain network model and would simulate the brain network model, we would find traveling waves from the temporal regions to the frontal and the occipital regions, so from low in strength to high in strength regions. And this was a very typical brain network model, and initially equipped at the nodes with very simple Kuramoto oscillators that were coupled. So in each node was this oscillator. Also, the time delays were considered. There was a transmission speed of 3 meters per second implemented. And with these simple brain network models, simulations have been run. And as you can see with the naked eye, this is an example simulation, traveling waves are emerging in this brain model. And it's a little bit difficult to recognize and to say, are the traveling waves now really following the in-strength gradients that have been determined in the connectome? So this was also statistically analyzed. The flow potential of traveling waves has been determined, and with this, the sinks and the sources, and we see that at the area of a low in-strength gradients in the temporal region, there's a source, and at the frontal and the occipital regions, there's a sink. So there are the flow, the wavefronts are following the in-strength gradients. So now as a next step, the question was, is this also the case if we not just couple the oscillators with this structural connectome that has in-strength gradients, but more physiologically grounded neural mass models? And the choice was to use the Janssen-Ritt model that consists of pyramidal neurons that get input from the connected network and also input from excitatory and inhibitory interneurons. And as you can see here, the pyramidal neurons are also driving the interneurons. So this is a little bit more complex neural mass model that is also capable of producing typical rhythms as we can see them in the EEG. And we now replace the Kuramoto oscillator in each brain region, in each brain parcellation with this Janssen-Ritt model. And the outcome, the simulation outcome is very similar. Here, again, an example. And yeah, you always can spot it that there are traveling waves that follow the in-strength gradient of the structural connectome. And this was also statistically evaluated, and at 90% of all time points, the traveling waves were emerging during the simulation, and in 80% with the Janssen-Ritt model, they followed the in-strength gradient of the structural connectome. So here we see again the in-strength gradient, and in our model we also looked at the frequency, the efficient frequency, so the intrinsic frequency was 10 hertz, and based on the in-strength gradients, in the intrinsic frequency that has been implemented in the model was actually modified. And what you see here is the effective frequency. And we see that the in-strength gradient modulates effective frequency such that in frontal and occipital areas, we have lower frequencies, and in temporal areas, we have higher frequencies, just because of the in-strength gradient of the structural connectome. So now, of course, the brain is a little bit more complex, and often you see several traveling waves at once, also at different scales. So the question was now, can this also be explained? Also, we know that the frequency gradients can actually be opposing for different frequency bands, for example, the alpha and the beta frequency band. So what we did next was we decomposed the single structural connectome in different structural connectome components. Yeah, here you see component 1, component 2. And we postulated that the different components can lead can shape different frequency gradients and different frequency bands, which then could explain opposing traveling waves that are co-occurring in the brain at the same time. So we now created such subnetworks by non-negative matrix factorization. And here you see two examples, so the alpha subnetwork and the beta band subnetwork. So we have structural connectome component 1 and 2, and if you add them up, you would get your initial full structural connectome. But here we see that the in-strength gradient is actually for the two components divided into two opposing in-strength gradients. Yeah, for the alpha band, we see a high in-strength frontally, and for the beta band, we see a high in-strength occipitally. So if we now run simulations for the two subnetworks, structural subnetworks, we see that we get opposing traveling waves in the same brain network model, so to say, consisting of two structural subcomponents. And we see that the traveling waves in the alpha band travel from occipital to frontal, and the beta band from frontal to occipital. So they're opposing. Okay, and yeah, this is already the summary of the first study that I wanted to show you more in depth. So the in-strength gradients exist in the human connectome. In-strength gradients shape the effective frequency gradients and thus direct cortical traveling waves. So with the study, we have found a candidate mechanism how the traveling waves are being generated in the human brain. And before I now switch to the next study, I will give it a little break, and I will now stop or pause the recording, if I find the button, and give you the opportunity to Okay, so now we continue with the next study that I want to show you a little bit more in depth, knowing that you have actually, oh, I see. This is bad. Actually, this is not correct. You will see in a moment the actual authors here. The first author of the study that I'm going to show you is Michael Schörner, this guy, and you will meet him, I think, in 2 days for the first time, where he will introduce how to compliantly process such sensitive data like brains. And then 1 day later, together with Luca Brandt, he will probably also go in depth of the study. But I like the study very much, and therefore I also want to inspire you with it already today, But if you have then in-depth questions for the study, you have the opportunity to directly talk to the person who has implemented the model and knows all the details far deeper than I know them. So what I like here in the study is that it is going beyond dynamics. What has been done is, again, we used Human Connectome Project data to construct individual virtual brains, about 650 we had here in this case. And then we plugged into these 650 individual brain network models, such a circuit model that has been investigated before in detail in isolation and has been shown to be capable of mimicking decision-making and working memory. Yeah, and you see that it consists of 4 populations that are mutually inhibiting. So the blue connections are inhibitory, the red ones are excitatory. And 2 populations get input from the frontal cortex, and the other 2 competing populations get input from the posterparietal cortex. And if we plug them in the brain network models, we have seen that the performance of the decision-making, the accuracy of decision-making varies. And we have compared then the accuracy or performance with the actual performance of the individuals from whom we had the data to construct the brain network models, and from whom we also had actually cognitive performance data. And the models, the fact that we encapsulated this behavior in mathematical models enabled us then to understand what is the source of variance in performance in these individuals. And I will explain what you see here in detail in later slides. Again, there's a good likelihood that Michael Schörner, the first author, will repeat a little bit on this, but I think this is quite a complex study, and this is to your advantage to hear some parts twice. So let me start with the fact that you are well aware of that decisions can be made fast, and they can be easy, like knowing that we have to stop at a traffic light that turns red, or the problems that need decisions can be difficult, and then solving them, taking the decision can be slower. And this has also been formulated and observed by the Nobel laureate Daniel Kahneman, who wrote the book Thinking Fast and Slow. And he postulated 2 systems. System 1 operates automatically and quickly with little or no effort and no sense of voluntary control. And system 2 allocates attention to the thoughtful mental activities that demand it, which is often associated with a subjective experience of agency and choice. Yeah, so the easy system, quick decision, and the demanding system that requires us thinking, yeah, which we notice, which is a thoughtful process. And the thinking capability is measured in cognitive psychology with so-called fluid intelligence tasks. And the Human Connectome Project also has used this task to do this with their participating human subjects, from which they also collected imaging data that we use then to create these digital brain twins. So these individuals did the fluid intelligence task, which consists of 24 such patterns, and the task was to complete the empty space and to find the hidden rule. And the 24 tasks have increasing difficulty. So it starts easy. So this is one of the easy tasks, and it ends very difficult, so that several participants are not able to solve the task and find the hidden rule. So this is called the Raven's progressive matrices task, and this is the task that underlies the score of fluid intelligence that is determined for the study participants. So now I pointed you to the book of Daniel Kahneman, the Nobel laureate, who postulated a fast and a slow thinking system. And he actually just came to these conclusions by observing the behavior of humans. And we have now the possibility to not only observe the behavior, but also to model it through very complex mathematical models. And this is what we did here. We built such a multi-scale model where we have the large-scale network, and then this is at a little bit finer scale where we have more detail in a circuit model that is plugged in the frontal and posterior regions of each individual brain twin. And just to give you a little bit the background of this model, this is actually, these are illustrations coming from the paper of Murray et al. Yeah, he has characterized this little circuit model in isolation in detail. You see here two competing pairs of neurons. Group 1 is labeled here A, group 2 is labeled B. And these two groups are competing, A versus B, and they are mutually inhibiting. It's a winner-take-it-all circuit model where if you put input into one of the populations in the posterior parietal cortex, then they will start a race, a competition, and the population which reaches the threshold, here you see the threshold in the prefrontal cortex first, will then be the winner, and the decision would be then made for this population, A or B. So if we look here at the traces, we see there are two different colors, easy and hard. So easy means that the contrast of the inputs going in the two competing populations in the posterior parietal cortex is very high. So the difference of the inputs is high. For example, much goes into A and only a very tiny current goes here. And then very quickly, the population that receives the larger input will ramp up. And the same happens in the prefrontal cortex. They're one team, they're mutually exciting each other, but they're also inhibiting the competitors. And this will suppress then the competing population, and they will decrease their activity. So if the contrast is high in the easy condition and the purple condition, there will be a quick ramping up and a quick reaching of the decision-making threshold in the prefrontal cortex. If the contrast is low, so the inputs in both populations is very similar, and there's only a small difference, then this takes longer and also can lead to decision-making errors. So despite the input would be higher, for example, to population A, it could happen that in this dynamic winner-take-it-all cycle, because of the noise on top, the other population would win, and this would be then an incorrect decision. I show you an example later. Again, some illustrations from the work of Murray et al., who have looked at the circuit model in isolation in detail. And here they are looking for the capability of the circuit model to mimic working memory. So we have talked about fluid intelligence, which means the capability of thinking, thoughtful decision-making. And the working memory is linked to cognitive flexibility. Can you switch quickly? If you have stored a working memory trace, for example, coded by this neuronal population here, and then there comes the next stimulus. Is the next stimulus capable of disrupting this trace? In this case, not. The population maintains the high activity level. And the working memory content would be maintained here in this example. In this example, where the working memory content is still more vulnerable and the next stimulus, the distractor, comes quicker, we see how the working memory trace would be disrupted, and then the new working memory trace would be maintained. And this would be cognitive flexibility. There would be switching, a quick switching of between the content in working memory. So this illustrates how this little circuit is capable of mimicking decision-making, also working memory in humans. So now, as mentioned before, we can plug this little circuit model in brain network models that are informed by the data of individual study participants. And we did this, but we also did in this study another innovation. In brain network models, usually the neural mass models, often consisting of an excitatory and inhibitory population that are mutually coupled, they are connected via long-range excitations with other brain regions that then also are equipped with such a network of excitatory and inhibitory populations. And this would be another region. So this long-range excitatory connection is typically done in brain network modeling. What was new was that we also included feed-forward inhibition. So there's not just the excitation, but there's also excitation going in the inhibitory population, which leads to feed-forward inhibition. So the effect would be inhibitory. And with this, we can actually create a ratio of long-range excitation and feed-forward inhibition. And this looks then such. So for example, here we have a strong feed-forward excitation and a relatively weak long-range excitation and a relatively weak feed-forward inhibition. And here we have the contrary. We have a weak long-range excitation and a relatively strong feed-forward inhibition. So this would be a low excitation inhibition ratio. This would be a high excitation inhibition ratio. And Michael Schörner, the first author of this study, showed that actually this ratio of excitation, long-range excitation, feed-forward inhibition is a useful mechanism or tool to systematically modulate the degree of synchronization between the coupled brain regions. And this can be seen here. So this is the degree of synchronization, or also called functional connectivity. And this is the excitation inhibition ratio. And this can be systematically varied with a low excitation inhibition ratio from a anti-correlation via a medium excitation inhibition ratio where we have no correlation, and then a high excitation inhibition ratio where we have full correlation or synchronicity. So anti-synchronicity, no synchronicity, and synchronicity. And this can be systematically varied by the excitation inhibition ratio. And this was a nice insight, actually. And Michael Schörner now used this mechanism and the learning rule to systematically, as you can see here, change the excitation inhibition ratio between all the regions. This is the functional connectivity matrix of the empirical data. And this is a simulated functional connectivity matrix. And by applying this learning rule, the functional connectivity can be fully reproduced in the model, as you can see here. So we have a very high fit between or correlation between the empirical and the simulated functional connectivity at the end of the fitting procedure. And this was important because it's exactly this structure of synchronicity and asynchronicity that shapes the cognitive performance in human brains, as we will see in a moment. So now, if we look into the model that has been tuned, as I have shown you in the previous slide, then we found that the functional connectivity in the empirical data and the simulated data was nearly the same, actually, was anti-correlated with the input the current that drives the related nodes in the model. Yeah, the currents cannot be measured in the human brain, but the currents that drive a node in a model can be, of course, quantified. And this was one of the advantages, that we have now encapsulated all the information in a mathematical model where this input current could be determined. So we found that the input current that drives a node in these tuned models was anti-correlated with the functional connectivity. And on the other hand, the functional connectivity was correlated with the correlation of the currents that was driving the two coupled nodes in question. So positive correlation between functional connectivity and input correlation, and the inverted correlation between functional connectivity and input strength or amplitude. So in other words, when we change systematically the long-range excitation versus feed-forward inhibition ratio, then we can not only or modulate the functional connectivity. So here we have again the excitation inhibition ratio. See how this changes systematically the functional connectivity, but not only that, it also changes systematically the input correlation that drives in the model the node, and it also systematically modulates the input strength or amplitude that puts currents in the model node. So now, if we plug this winner-take-it-all circuit model that I've shown you previously in these two brain regions of our 650 human connectome project-informed brain models, what happens? How does it look like? We used in the brain network model the reduced Wang model, and these are the equations of the circuit model, the winner-take-it-all circuit model, and as you can see, they are very similar. So the two models are very well related and also actually developed by the same group, by Wang et al. So what we see here are the input currents for the excitatory population and the input currents for the inhibitory population. They are translated in the reduced Wang model into firing rates for the excitatory population and inhibitory population. So R are the firing rates, excitatory, inhibitory. And these are translated into synaptic activities, again, for excitatory populations and inhibitory populations. And in the circuit model, we have very similar equations, and we have also a noise term for the competing populations, but the noise terms are now replaced by the input that comes from the network in which the circuit model is embedded. Yeah? So for some of the population, it would be the input from the purple colored nodes, and for the other circuit populations, the input from the turquoise colored nodes. So the noise term is replaced by the sum of the activities of these colored nodes. Yeah. So with this, we couple the brain network model with the circuit model, which previously has only been investigated in isolation. And how does this now look like if we simulate the circuit model? And how can we measure the performance of the decision-making performance of the circuit model? So this is just one trial of a simulation, and the color of the traces reflects to which of the four populations they belong. Here we see that the evidence, the current that drives the pair A is a little bit higher than the evidence or input current that drives the group B. And this means the correct decision here would be A. So A should be the winner. If B would be the winner, it would be an incorrect decision because the evidence is higher for A. And in this example, we see that indeed the blue population reaches the threshold, the maximum, and the green population is suppressed. So the decision would be for blue A. This would be a correct decision. So now we can do this again, and this would be an example since the contrast is very low and there's noise in the system. As mentioned before, there can also be incorrect decisions. And here, despite the evidence being higher for A, the decision would go to B, and this would be therefore an incorrect decision. And this ratio would be the performance. Yeah? So the more correct decisions, the less incorrect decisions, the better it would be the performance in our model. And this can be repeated. We have repeated it here 1,000 times. And here you see such a ratio. So in this example, we would have 790 correct decisions and 210 incorrect decisions. And we did this for our 650 individual brain network models and found that the brain models of smarter subjects made more correct decisions and took also more time for decision making. Yeah, so models of smarter individuals measured by the fluid intelligence score had a better performance, simulated decision-making performance in our model. But, and this was unexpected, they also took more time to come to their decisions. So here again on the x-axis, you see the fluid intelligence score. The higher the intelligence score, the longer was also the decision-making time, the reaction time. But this time we didn't have the data, so this was unexpected. We were a little bit surprised to see that the more fluid intelligent people took longer in our simulation to produce a response. And then Micha contacted Diana Bark, who is the contact for the Human Connectome Project behavioral tests or the Human Connectome data study. And we asked for the reaction times in this progressive matrices task. And she really provided the reaction times, and then we were able to analyze them systematically. So as mentioned, the progressive matrices fluid intelligence task consists of 24 individual pattern completion tasks, starting with an easy and then increasingly getting more difficult. So here, what you see here is the analysis of just the first task for all the subjects, the most easy task, easy problem, question 1. And here we found that the higher the fluid intelligence or also the general intelligence score, the faster was the reaction time. So more intelligent, faster reaction for an easy task. For the last question, question 24 of the progressive matrices fluid intelligence task, this is a hard problem, difficult to solve. We found the inversion. So the higher the intelligence, the lower was the reaction time. And this was also analyzed here for all the individual tasks. So here we have only question 1 and question 24. Here we have them all 1 to 24 group averages and the correlation between intelligence score and reaction time. And we see until question 8, it was an inverse correlation. The higher the intelligence score, the faster reaction. And then from question 9 on, suddenly this inverted. So it got difficult. And then the reaction times were longer for those who scored higher in the fluid intelligence task. And this was also the same for the individual subject level. So what we also found was that the slow solvers, who took more time in reacting or deciding, had a higher synchronization, higher functional connectivity in their data. Okay, I need to drink something. 1 second, please. Just find. Okay, now it's better. So functional connectivity, degree of synchronicity is related to the reaction time. And what we see here now is the phase space. So this is now the nice opportunity that we have with a mathematical model to look at the phase space and to see how does the amplitude that goes in our model node, yeah, this is shown here, the amplitude that drives the node in the posterparietal cortex, relate to the number of correct decision or ratio of correct decisions? How does this amplitude that drives a node relate to the integration time or the reaction time? And how is this actually explainable? Yeah, and this can be understood through the phase space, which I show you in the moment in more detail, just to give you already a first glimpse. So now we go in the details and to understand this better. We have here now 3 different scenarios: low synchronicity or low functional connectivity in the brain, fast decision making, but also less correct decision making, medium synchronicity, high synchronicity or functional connectivity, slow decision making with a high accuracy. These are our simulated data for the 3 scenarios. And we see if we have low synchronicity or functional connectivity in our brain model, we have a high fraction of incorrect decisions, 210 of 1,000. If we have high synchronicity in our brain model, we have a high performance with a low fraction of incorrect decision, 41 of 1,000. What we also see is that the decision making in low synchronicity is faster. Yeah, already after 0.5 seconds, the threshold is reached in most cases and the decision has been taken. For high synchronicity in the brain, it takes longer. So it takes more than 1 second until the activity is ramping up and the decision is taken. And this is what the data have shown us that I have shown you previously, but now we want to understand why is that. And this is where the model helps us. So we now look at the phase space. The phase space is spanned by the activity of the competing populations in the posterior parietal cortex, population A and population B. And the little balls are so-called fixed points. The little arrows are flow fields that show where the system is pulled. And we see now that when we start our simulation, and we color here the trajectories of the different simulations according to where the decision has been taken, either for A, then the trajectories are colored in green, or the system ends up in B, decision B, then the trajectories are colored in red. And we see the 2 fixed points here, and we see that relatively quickly the system is pulled to one or the other. Yeah, this is shown here after only 0.5 seconds, the decisions already have been made, and the system ends up in one of the 2 attractors and fixed points. If you look here in the high synchronicity or high functional connectivity scenario, we see that actually the configuration in the phase space is different. Again, it is spanned by population A and population B in the posterparietal cortex. And we see that actually here we have a fixed point in the midline, yeah, that keeps the system trajectories in a balanced state. Yeah, they cannot go to either of the 2 attractors or fixed points. They cannot take decision A or decision B. They are stuck here in the middle. And this means the system remains balanced, and this gives the system more time to integrate the incoming evidence. Let's now continue. Okay, now if we continue our simulation, we see that after 1 second here, all the decisions have been made already, and quite a high number of incorrect decisions after a very short time. In the medium synchronicity, we now see that also decisions have been made. In the high synchronicity state, there's still this fixed point, and only now we see that the trajectories start moving a little bit away from the balanced state, from the diagonal, the balanced activity between populations A and B. And this means that the system here had already about half a second more time to accumulate the evidence and to average out the noise. And come to a better informed decision. And now we continue, and only after about 1.5 seconds, this little fixed point here has resolved. This is because of the feedback coming from the prefrontal cortex. This has been also analyzed in the publication. I cannot go in the details now, but basically if the activity ramps up in the prefrontal cortex, then after a while this fixed point is released, and this then leads to a better informed decision, which with a lower fraction of incorrect decisions. And this shows you how nicely such a brain network model can help us not only to understand the dynamics, for example, of traveling waves, but also how this leads to different cognitive performances. And this is, I think, just a summary slide, fast but faulty versus precise but slow decision making. We have looked into the empirical data, the large-scale model, and how the large-scale model then affects the small-scale decision-making model. And this is at the end of the second part of my presentation. I have a last part. I do not go so much in the detail of the last study, but I want to give you some other inspiration still. But I think I stop also here once more in case there are any questions.